So, today I welcome you all on the SWAM course on genetic engineering and its recent advancement. The topic of the talk will be the bacterial artificial chromosome and yeast artificial chromosome. So, now we are moving towards the very important tools of cloning and expression vectors in the process of genetic engineering. As we know, the genetic engineering is a series of process where we try to construct, we attempt to make a recombinant DNA technology, or we can say to create the recombinant DNA to use it for various applications, right. So, in this regard we see how these two type of vectors are useful for the genetic engineering applications. So, we say basically it is a artificial chromosome which can be said as a very important powerful tool for genome analysis, for making different type of genomic libraries, for cloning applications, for expression applications, for various type of genomic analysis. So, what are artificial chromosome has to be understood, and then we see these two type of artificial chromosomes. Let us see the way how we find out the artificial chromosomes. So, everyone here knows about the chromosome, and this is a prefix which has been given. Artificial means that it is the engineered version of the chromosome, right, and we can say it is a large engineered DNA molecules and it mimics the behavior just like the natural chromosomes. So, the size has been increased. Normally we try to construct various type of cloning vectors. We have attempted for the expression vector, and in the same series we see that how this engineered chromosomes can be designed in lab, which can be said as a artificial chromosome because it carries all the or we can say it will mimic the behavior of the natural chromosome. The idea is to design this artificial chromosome because normally the size is too large comparative to the any extra chromosomal material, and this are designed to carry the large DNA fragments. So, we are trying to construct such type of chromosomes so that we can further increase the uptake size of insert DNA, and this time when you make this version of the chromosome which is called as artificial chromosome, further the length of the DNA can be extended up to around 100 kilobases, and this is right, it can be used. Basically we know that this is the era where we are attempting to clone the genome of various organism to identify, to study the particular gene segment. The entire genomic regions can be cloned in, or the large gene clusters has to be cloned. This is the objective of your genetic engineering or the genome projects, where we try to clone the entire genomic regions for various kind of studies related to the gene and to create the genomic libraries. So, artificial chromosomes are made and they are very useful for cloning the large genomic regions, so that you can entire genome can be cloned and you can study the gene functions, the gene regulations, and you can preserve it, you can propagate it, you can store it in the form of genomic libraries. And to make the genomic libraries, certainly the requirement of the cloning capacity or the cloning limitation has to be overcome, and we certainly need to have such type of vectors where still we can increase the size of the insert DNA fragment. Next we see that these are the two type of chromosomes we are very much commonly in, now we are engineering it in, it is a designing in the your normal test tube such kind of chromosomes which is behaving like a natural chromosome. So, we have two versions here, one is the bacterial artificial chromosomes and another is the yeast artificial chromosomes. So, we see what are these two and how they act as a vector. So, bacterial artificial chromosomes are the first we see because it is used for cloning and manipulation of larger DNA fragments of DNA in bacteria. As we know, your contemporary, or we can say that very commonly used, the conventional DNA like plasmid DNA is there, bacteriophages are there, and you have prepared cosmids also for the cloning application for manipulating of the gene. Now, in this series we see that how the new development has come, which is termed in the name of bacterial artificial chromosome, because this has been derived from the bacterial plasmids, and we see that it has the host cell is the bacteria. So, these are created by inserting the foreign DNA fragment. This is a necessary essential step for any vector. So, we take the gene of interest, we insert into the bacterial plasmid, and then it is inserted or introduced into the bacterial or host cell, where it replicates and then write just like a bacterial chromosome. But what is the difference over here from your conventional cloning vehicle is that it can accommodate, right, the DNA size is exceedingly increased. Here you can clone up to 300 kilobases in size. So, just imagine that this limitation is how overcome in the form of bacterial artificial chromosome, and that is why it is a very useful vector for cloning the entire genes and mostly the gene clusters for its manipulation and for its cloning application. We see ki if you create the bacterial artificial chromosome, mostly they are the engineered version of the F factor plasmid, which is also called as the fertility plasmid. It has been derived from the F type of plasmid, and it carries the all the essential sequences, like the first thing is the Rep-E, right. It is required for the replication of plasmid, right, and this replication is also important to control its copy number into the the origin of replication is also equally important for its maintaining the stable copy number within the bacterial cell. And so far when you engineered it, obviously this new construct which is called as artificial chromosome is also their copy number is depending on the replication feature like Rep-E, which is present in the bacterial artificial chromosome. Now, the next two important thing is the par A and par B. These are the use for partitioning the fertility plasmid into the daughter cells. So, you know that this is the bacterial cell division is an important part, and F factor plasmid is called to be the fertility plasmid because it is helping the bacteria for the conjugation, for the transfer of genetic material from one cell to another cell. And when it gets divides, though we need that certainly that once it division will be started, their partitioning is important, and this partitioning is equally controlled by the par A and par B sequences, which is present also in the artificial construct. It provides the stability, so that it is not going to be lost in the process of division, and it has to be maintained throughout the in the dividing cell into their daughter cells. We have selectable marker. So, these are the features which is present in the F factor plasmid, which has been added in the form of selectable marker, which confers some type of resistance and which allows the researchers to select the bacteria that have taken the bacterial artificial chromosome. So, multiple cloning site is one of the important feature, as you know that in addition to the conventional plasmids, we have seen the other features of F factor, but the common features are like presence of selectable marker, multiple cloning sites should be available, so that the foreign DNA can be inserted into these unique sites where the DNA of vector is digested to take or to insert or to link the gene of interest. And one more important sequence is essential sequence is the origin of replication. So, it has the few new sequences from your conventional plasmid, that it is also having the copy number controlling element, it is also having the partitioning stability element along with the origin sites, multiple cloning sites and your selectable marker sites. So, F factor plasmid is important, and this is like based on, this is the engineered version of the F factor plasmid, and this factor plasmid is called as a fertility plasmid because it is having this natural ability to mobilize from F plus donor bacteria to F minus female bacteria, and that is why it is called to be the fertility factor. It takes the genes from one cell to another cell by the process of this gene transfer is happening with the help of F factor, which is also called to be the conjugation, and this conjugation tube is made by the pili which is present on the surface of bacterial cell, and through this the gene transfer is naturally happening by the process of conjugation with the help of F factor plasmid. So, this is the engineered version of the F factor, which where we have added new sequences to act as a cloning vector. F factor having its own replication, it can control its replication, right. So, it is having two origin of replication. So, you see that in the extra chromosomal circular DNA in the form of F factor plasmid, it is having the ability to for the bidirectional replication, for the unidirectional replication, because it is controlling the gene transfer. So, it has having the two, it is a natural presence of these two type of origin which is present in the F factor plasmid, like ORI V site, ORI S site for its different direction of the replication, and it also has a gene that regulates its own DNA synthesis. So, this is very important. It can control its copy number, it is keeping at a low level, and genes are also present which regulate the partitioning into the daughter cells after E. coli divides. For example, here we took the E. coli. So, here when the E. coli divides, the new daughter cells are made, and how these new daughter cells, the F factor is distributed or partitioned is also regulated by their gene which I have told just now, that is about the PAR A and PAR B. It is having RAP sequences which can control its replication capacity, so that it can control its copy number within the host cell. So, this is a very schematic important diagram of one type of bacterial artificial chromosome, which is showing all the necessary elements or genomic sequences to be present in the bacterial artificial chromosome. So, you can see that it is having the origin of replication sites, it is having the PAR A and PAR B sequences for its partitioning stability, it is having the RAP E sequences, RPE kind of sequences which controls or regulates its DNA synthesis, so that it can control its copy number. It is also showing the selectable marker gene and also showing the sequences of unique restriction enzyme, where the back is open to take the large DNA insert. Actually, how we create the back as a cloning vehicle. So, these are the steps. So, first we have to isolate the DNA. When you construct or you start the cloning purpose, first of all the ones you have to isolate the genomic DNA from the organism of interest. Then the genomic DNA, once you isolate it, then you cut into the smaller pieces. You have the tools like you have the restriction endonucleases enzyme to isolate the gene of interest from the isolated DNA or the genome. Then you have to ligate into the vector. The fragmented DNA or the isolated gene of interest is then linked or ligated to the back vector, which is a kind of modified plasmid which contains all this. F factor has been modified to added, it has been added with the selectable marker, cloning sites, right, to insert the DNA fragments. So, this is the site where you insert the gene of your interest, you ligate it into the bacterial artificial chromosome, and this is the T4 DNA ligases, the enzyme you add in the reaction mixture, so that you can add the new DNA into the bacterial artificial chromosome, because it is a natural mechanism of conjugation which helps the F factor to be utilized as a bacterial artificial chromosome. And to get that, to achieve that, all the ligated mixture is then transformed. That is how you introduce the back into the competent bacterial cells, means the cell is ready to take the new DNA material. And this is the selection process. Like once you have transferred it, the next step will be the transformed bacterial cells. You have to select the cells or the host cells containing the back clones, and for that you are plating that cells into the selective media containing the antibiotics as a selectable marker. So, if it contains the antibiotic, it will kill the non-transformed cell, and only the transformed with the appropriate back vectors carrying the vectors will be able to survive in the form of colonies. So, this is the selection is based on the presence of its survival on a media containing the antibiotics. So, only the cells which are having the back clones will survive over there, and rest of the non-transforms are killed. And screening is done once the colonies are formed. Then you have a various techniques you can take the you can use it to identify the back clones for identifying that it carries the DNA fragments of interest or not. So, you can apply various type of hybridization techniques, right, PCR amplification methods, right, so that it will the next step will obviously the you have got up to the selection that it is the host cell is carrying the back clone, but after that the very important thing, the successful event has been done is to get that information that whether the back clones containing the gene of interest or not, and to identify the clones containing the gene of interest. Once again you have the various techniques by which one can apply and one can use it like hybridization and PCR designed to the flank of the gene of interest, and it will amplify those region only, and which will give you the clear screening of the back clones containing the gene of interest. So, these are the steps in the form of figure. You can see that this is the cloning site. So, first on the top you can see this is a back vector carrying all the necessary elements, genetic elements to be act as a bacterial artificial chromosome, and at particular its restriction site it is opened, and the large foreign DNA here you see this is not a small size, it may be up to 300 kilobases, and this fragment of large foreign DNA is inserted into it, right, and the whole recombinant, this is the first recombinant, by what I said is the recombinant vector is then electroporated. Electroporation is the process by which you introduce the, it is artificial method, you can create the pores on the bacterial cell membrane, so that you can introduce the foreign DNA or the recombinant back clone into the vector, and then you start the selection of chloramphenicol is a kind of antibiotic which we have used as a selectable marker genes. So, the colonies which appeared on the plates containing the chloramphenicol will help us to identify that this is the recombinant or we can say these cells are recombinant, they carry the recombinant backs, and you can easily identify the recombinant from the non-recombinant on this specifically designed screening or the selection plates. The next type of artificial chromosome which are going to be understand is the yeast artificial chromosome. So, we can say it is a human engineered DNA molecule, and it is used to clone the DNA sequences in yeast cell, and we see here there are it has been engineered because to increase the size of uptake of the DNA insert. Now, you can see now it has moved from, it has been extended from 300 kilobase pair to somewhere several thousand, like up to 1000 kilobases of the DNA insert. So, it is YAXR or the yeast artificial chromosome can take the foreign DNA insert into the yeast chromosome. Now, you see that when you define the chromosome, you need certain type of sequences to be present, and that is what we do in creating the yeast artificial chromosome. And here we see the important feature of the chromosomes are present like centromere, telomeres, and apart from that those features which help us to identify the clone, that is the selectable marker. So, this is particularly useful in studying. So, this has been created mostly for study of the eukaryotic genomes, say for example human genome. So, if you want to make the genomic library of the human genome or the cloning of the large gene clusters of the human for study, for its regulation kind of study, for its analysis. So, we need this kind of genomes can be studied very well in a eukaryotic host system. So, this has been specifically designed to study the eukaryotic genome. The important components of yeast artificial chromosome, the first one to understand, when you create it artificially, you identify certain segment which has to be added in the engineered version. So, first we see that it is showing the presence of centromere. Centromere is a very important sequence which ensures the chromosomal segregation when the cell is dividing at the phase of division, in the division process between the two daughter cells, right. In this process, the centromere is very important for the partitioning or for the segregation of chromosome. We know the importance of centromere. In this designed version, we see that the important sequence to be present will be the centromere, and then there is a yeast telomeres which are present at the end of chromosome, and telomeres have their important functions, like it is providing the stability to the end of the chromosome, and also this is important to sequence accurately the ends of the chromosome. So, that is why the telomeres are present. Then you have the selectable marker which confers the resistance property or some other such selectable property in the microorganism or in the host cell. So, now the next one, as we know, for any vector, the next important feature is the multiple cloning site. It is the region of the DNA sequence where it contains the sites for the restriction enzymes, and this is the site where one can digest the vector and it can insert the foreign DNA into the yeast artificial chromosome, and origin of replication is also important for its independent replication. So, here you see the important features are present, and once you identify, once you define the chromosome, artificially you can create such chromosome by isolating the components, each such components like telomeres, centromeres or origin of replication by the help of recombinant DNA technology, and in test tube you can join this sequence once again to create artificial chromosome. So, here we see that how the YAC or the yeast artificial chromosome has been created, and this is mostly it is derived from the plasmid PBR32, where we have inserted such sequences. We have designed it in vitro condition by the help of recombinant DNA technology. So, it is showing the marker sequences, centromeric sequences, ori sites, telomeric sequences. So, all the units or the segments are present in this vector, which makes it just like a natural chromosome, and that is why they are called to be the artificial chromosome. So, before being used in the cloning, the vector is propagated as a simple bacterial plasmid, and it is having the all the important cleavage sites, right, and you have the telomeric sequences, right. It has having the other important sites which is like selectable marker sites are there, and it is having the polylinker sites or the multiple cloning sites are there. So, these are the important features of the yeast artificial chromosome. So, as you know how you start the any cloning experiments, you have to isolate the genomic DNA, right, from where you have to cut it into a suitable DNA size or the fragment size which has to be cloned, and this genomic fragments you have the technique to separate it based on the size like of the electrophoresis. So, it is giving a little bit detail of how it can be used as a vector. So, this is a step of the recombinant DNA technology where you actually do the cloning. So, these fragments are isolated from the genomic DNA, then they are ligated into the yeast artificial chromosome, and finally, then once the recombinant eggs are prepared, they are transformed or introduced into the yeast cells. So, this is the kind of steps. So, you have the BAM-H1 sites, EcoR1 sites, you have the selectable marker, you have the centromeric region, this is all present, and you take one of the enzyme like BAM-H1, when you use it, you digest it, the chromosomal DNA or this artificial chromosome which is circular in form, it becomes linearized and it will create like 3 arms, right, and this 2 arms which one of the arm which is flanked by the BAM-H1 site is discarded, and other 2 arms in between that you have the site where you can attach or you can link the EcoR1 digester site is used to create these 2 type of the arms. And then finally what will happen, here you have to in the polylinker site or this restriction enzyme which has opened the yeast artificial chromosome, exactly in the same site you insert DNA or the foreign DNA of your interest, and then they are ready to be injected into the or to be introduced into the yeast cell. So, here you see that EcoR1 digester site creates the 2 arms. So, you can see here it depends on which enzyme you are using. If you are applying the BAMH one, it will create 3 DNA fragments. If you are using it the EcoR1 digestion, then you have the left arm and right arm. So, each arm is flanked by the telomeric sequences. In between this, up downstream to the centromere, right, you add downstream to the centromere, in between the centromeric sequence and the telomeric sequences, the DNA of the interest is attached, and then it makes the recombinant artificial YAC artificial chromosome, and then this is transformed into the yeast cell, which is normally in the form of protoplast, because protoplast form can be easily used to introduce the new or the external kind of chromosome into the yeast cell. So, you do the enzymatic digestion of the cell wall and you create the yeast spheroplast, and this yeast spheroplast is then used to introduce the YAC chromosome or the recombinant YAC into the yeast cell. So, this is how we see that if you make this kind of help of the YAC, you can further increase the sizes and you can have the selection and screening process where you can use the different strategy to isolate the cells carrying the or containing the yeast artificial chromosome. And the very good advantage over here is, like if you are using the large size genome for its library application or for its study application, you need the number of clone is very less. So, you need that instead of several thousand clone, if you increase the cloning capacity, you have to handle less number of clones. So, that is the major advantage when you increase the insert size, you increase the cloning capacity, the number of clone you have to handle will be less, and it is a significant reduction in the number of clones to be handled, which is easy to maintain for a longer period of time. That is why we purposely made engineered version of this chromosome. First is the bacterial artificial chromosome and the second one is the yeast artificial chromosome, and you can clearly see the difference in the host cell. One is propagated in the bacteria and another on the yeast, and obviously in the yeast means you can study the eukaryotic genome, insert size changes are changed. In the bacterial artificial chromosome, the cloning limitation is up to the 300, but it is very good with respect to the conventional plasmids, and here you can see further increase in the cloning capacity with several hundred kilobases. Stability, if you see in the bacterial artificial chromosome, is less because there are the chances where there is a chances of the rearrangement will be there, but when you talk about the yeast artificial chromosome, it carries all the necessary sequence for its stability, like it is having the telomeric sequences, centromeric sequences. So, its segregation is also just like a normal chromosome or natural chromosome. Bags are made on the plasmid, it is a derived plasmid derived, and it is always a extra chromosomal replication, but here the yeast artificial chromosome, it replicates like chromosome and it segregates also just like the natural chromosome. So, it behaves very much close to the natural chromosome because you have added all the necessary genomic sequences which convert any DNA fragment into the chromosomal form of eukaryotic nature. Cloning method is, as you know, in any cloning we do, we insert the DNA into the plasmid, and here even the cloning can be done by homologous recombination. So, that is also a possibility because the features are same, and here you can see that there is also a chances of homologous recombination with the host yeast genome, and the novel property can be derived from there. Markers are, you have a choice, like instead of conventional antibiotic resistance, you can use the auxotrophic mutants or the nutritional markers also. Like you can see, you need to add some specific nutritional supplement to find out the non-recombinant, or in between you can differentiate between the non-recombinant with the recombinant by having the auxotrophic mutants. Means if certain nutrients are not given, the non-transformant will die, whereas the transformants are positive and they can still grow in the case of auxotrophic conditions. So, nutritional markers are also added apart from the antibiotic resistance. And here you may see the applications are wide. You can use both kind of chromosomes for making the libraries, for even the gene expression analysis, for making it for the functional. It is not exactly about the differences, it is also about their common applications. When you study in a larger size, you can, even if you are able to do the cloning of large gene fragments, of course an obvious reason will be to express the gene, to study its function. So, these are the important other area of the genetic engineering which can be applied easily in the case of such artificial chromosomes. So, this is very important to understand why we have engineered the bacterial and we have created artificially the artificial chromosome. The major reason is to increase the insert size of the DNA. So, as compared to the plasmids, as compared to the phages, and phages, when you take, the complexity comes where to particularly the in vitro packaging systems are there. So, there are some complications related to the each type of application of the vectors, and then you have designed the cosmids also. Cosmids are also, it is a combination of the phage and plasmid, where you can make the chimeric kind of DNA, right, or the recombinant DNA. First you create between the plasmid DNA and the phage DNA just to increase the capacity. So, from the plasmid you can, if you compare the plasmid cloning capacity to the cosmid capacity, the capacity is further increased, but it is still a major hurdle or the technical barrier where you, once you want to study the eukaryotic chromosomes. So, eukaryotic chromosomes are sizes are varying and they are very extensively large. They have several hundred kilobase pairs in length, and we need certain type of vectors which can take this large gene clusters. It can clone this large gene cluster, it is able to manipulate this, and also it is able to express this. And for these reasons the yeast artificial chromosomes are very important. If you say, for example, when you take the this yak is basically derived from the PBR 322, which is having the capacity around 20 kilobases. But when you convert into the yeast artificial chromosome, the same plasmid, then this engineered version is carrying the 1000 kilobases in the length. So, just imagine there is a almost 100-fold change. So, if you increase the large in the cloning capacity, the clone number is decreased, and when you decrease the clone number, this is a very essential step in the gene cloning. If you are working with the genomic library, it is easy to maintain and handle, right. It is a very reliable method where you can control the clone size for its maintenance in the form of genomic libraries. So, I thank here. I hope you will be able to understand with these two type of advanced cloning and expression vectors. Thank you.